I am Celine Hilda Mary going to present about the radioimmunoassay. Immunoassay is a technique that uses antigen-antibody complexes. An antibody-antigen complexes is known as an immune complex. Immuno refers an immune response that causes the body to generate antibodies. Assay refers to a test. Immunoassay is a test that utilizes immune complex when antibodies and antigens are brought together. An antibody is a protein that is produced by the body in response to an invading substances. Antibodies are produced as a part of the body's immune response to protect itself. An antigen is a substance that induces an immune response. For example, the drug is an antigen that binds to the antibody. An immunogen is a substance that elicits immune response. Example, drug protein conjugate. An analyte is anything measured by the laboratory test. In immunoassay testing, the analyte may be either an antibody or an antigen. Immunoassays utilize one or more selected antibodies to detect the analytes of interest. The analytes being measured may be either naturally present in the body, such as thyroid hormone, the body produces but are not typically present, such as cancer antigen, and also they do not naturally occur in the body, such as an abused drug. This is the structure of antibodies. Antibodies are produced by B lymphocytes in the biological system. Most common one is the immunoglobulin G, that is IgG. IgG is a protein composed of two main structural and functional regions, such as FAB region and FC region. FAB region contains the antigen binding site that varies among different antibodies. FC region of constant structure within the antibody class, that is the fraction of catalyzed crystallized. Next, radioimmunoassay. Radioimmunoassay is an elegant technique in the analytical chemistry. If substance to be analyzed in a very low quantities in the order of micrograms, nanograms, the conventional methods like gravimetric and calorimetric methods may not be used. So RIA finds extensive application in this case of many substances which are present in the trace amount of in blood. History. RIA was developed in 1959 by Rosalind Yalow and Salmon Burson for measurement of insulin in plasma. For this, they have got a Nobel Prize. This is the principle of RIA. The amount of antibody per tube is kept constant. The amount of antigen added, that is both known and unknown antigens are added to the known antibody. The added antigen will be distributed between a bound and a free fraction. This distribution is governed by the association constant Ka of the antibody. That is K equal to sum of the concentration of antigen-antibody complex divided by concentration of antibody and concentration of the antigen. By using this formula, we can calculate the association constant. In the principle, competitive binding of radio-labeled antigen and unlabeled antigen takes place with the high affinity for the antibody. The labeled antigen is mixed with the antibody at a concentration that saturates the antigen binding sites of the antibody. As the concentration of the unlabeled antigen increases, more labeled antigen will be replaced from the binding site. The decrease in the amount of radio-labeled antigen with the bound specific antibody in the presence of the test sample is measured to determine the amount of antigen present in the sample. In standard condition, the amount of unlabeled antigen bound to the antibody decreases as the amount of the unlabeled antigen increases in sample. This is the picture that is with the known concentration of the antibody. The labeled form of the anti and the labeled form of the antigen is added and which binds to the antibody. Afterwards, the unlabeled form of the antigen is added. The unlabeled antigen replaces the labeled antigen, and as a result, the bound form of the labeled antigen is decreased along with the antibody. So, the measurement of the labeled antigen radioactivity is measured in order to detect the amount of unlabeled antigen present in the serum sample. This is the principle involved in the RIA technique. These are the reagents used for the in the RIA. A tracer that is labeled ligand is used, and a binder that is antibody which is the specific for anti-serum, and the third one is a separation system to separate the bound and free phases, and fourth one is a standard which is used in the highly pure form, and fifth one is a free human anti-serum is utilized. These are the reagents used in the RIA. That is, some of the radioisotopes are used commonly. That is, beta emitters, hydrogen tritium, as well as the carbon 14, and gamma emitters is commonly iodine 125 is used. What are the disadvantages of the reagents? Means they damage the ligand. The damage to the ligand may occur during storage. Health hazards are more in case of RIA. This is the specific anti-serum. The specific anti-serum that is a binder or antibody is prepared by injecting the antigen together with the fervent adjuvant into a suitable animal such as guinea pig, rabbit and goat. Molecule like thyroid hormones, steroids, drugs are not immunogenic, so they are conjugated to carrier proteins and polymer to make them immunogenic. And also a separation system is required because the bound fraction does not precipitate spontaneously at low concentration. Variety of procedures are available for the separation, and that is physical method, chemical method, as well as the solid phase system is utilized. In the physical method, filtration, chromatography, electrophoresis, charcoal, dextran adsorption are used. In the chemical method, organic solvents such as ethanol, PEG, salt, etc. is used, and solid phase system also utilized. A standard that is ligand or analyte should be prepared in the highly pure form. Drugs, proteins, hormones, etc. must be prepared in the pure form so they can be diluted. In case of protein hormones, the standard should be prepared preferably with the hormone from the species from which the serum is going to be analyzed. Next one is the ligand-free human serum. It is prepared by treating human serum with charcoal. It can also be prepared by collecting serum from volunteers in whom production of that ligand or hormone has been inhibited by treatment with an appropriate drug. Next is the general procedure for performing RIA analysis. A known quantity of antigen is made radioactive either by labeling with gamma emitters or beta emitters. This radio-labeled antigen is then mixed with a known amount of antibody for that antigen, and as a result, the two chemically bind to one another. The sample of serum from a patient containing an unknown quantity of that same antigen is added. This causes the unlabeled, that is cold antigen from the serum to compete with the radio-labeled antigen for antibody binding site. As the concentration of the cold, that is unlabeled antigen is increases, more of the more of it binds to the antibody, and by displacing the radio labeled variant and reduces the radio antibody bound labeled antigen to free radio labeled antigen. The bound antigens are then separated from the unbound ones. The radioactivity of free antigen remaining in the supernatant is measured. Separating bound from unbound antigen is crucial. Initially, the method of separation employed was the use of a second anti-antibody. This is the pictorial representation of the technique. The radio-labeled antigen is combined with the first antibody, and then the unlabeled antigen was added. After the addition of the unlabeled antigen, the radioactive antigen was replaced by the unlabeled antigen, and then in order to separate the bound antigen antibody complex, there is a second antibody is added. This second antibody precipitate the bound form of the antigen antibody complex, and the precipitate antigen antibody complexes with anti-immunoglobulin also takes place, and the radioactivity of supernatant was measured, that is free antigen present in the radioactive form is measured. This represents the concentration of the unlabeled antigen present in the sample. This is the procedure involved in the radioimmunoassay technique. And next we'll see about the advantages of RIA. Radioimmunoassay is a very sensitive technique used to measure the concentration of antigen without the need of a bioassay. It can also measure the 1 trillionth of a gram of material per milliliter of blood. It is structurally specific as antigen-antibody reactions are highly specific. It is an indirect method of analysis. It is a saturation analysis as active reagent added in smaller quantity than that of analyte. Next one is the disadvantages of RIA. It requires prolonged reaction time in days. As a consequence, highly diluted reagent is used. Radioactive iodine used is not a cheaper reagent. Possible health hazards due to handling of radioisotopes may take place. All the reagents must be added precisely. Limited assay range. Lack of direct linear relationship between analyte concentration and a signal response. Difficulty of automation and lengthy counting time is needed. These are the disadvantages of RIA. And next one is the uses of RIA. Narcotic, that is drugs, can be detected. Blood bank screening for the hepatitis virus. Early cancer can be detected by using RIA. Measurement of growth hormone levels. Tracking of the leukemia virus. Diagnosis and treatment of peptic ulcer. And research with brain chemicals called neurotransmitters can also be detected by using the RIA. Thank you.